Ladies and gentlemen, esteemed guests, please join me in welcoming today's guests of honor to the stage. Distinguished guests, ladies and gentlemen, online viewers worldwide, welcome to the 2024 Tang Prize Laureate announcements. My name is Grace Huang, your MC for today. From June 18th to the 21st, the Tang Prize Foundation will be announcing the laureates of the 2024 Tang Prize in Sustainable Development, Biopharmaceutical Science, Sinology, and Rule of Law. In today's press conference, the 2024 Tang Prize laureates in biopharmaceutical science will be unveiled. To begin with, I'd like to introduce the guests of honor that are here with us today. First of all, Dr. Zhang Wenchang, Chair of the Selection Committee in Biopharmaceutical Science. Dr. Gong Xingjian, Academician of Academia Sinica. And Dr. Chen Zhenchuan, CEO of the Tang Prize Foundation. And to kick off, I would like to invite Dr. Chen Zhenchuan, CEO of the Tang Prize Foundation, to give us the opening remarks. Ladies and gentlemen, please join me to welcome Dr. Chen.
Distinguished guests, ladies and gentlemen, on behalf of the Tang Prize Foundation and its founder, Samuel Yin, I would like to welcome you to the announcement of the 2024 Tang Prize in Biopharmaceutical Science. Established in 2012 by Dr. Samuel Yin, the Tang Prize is an international award that embodies the principle of sustainable development. The four categories of the Tang Prize, namely sustainable development, biopharmaceutical science, sinology, and rule of law, address critical issues for humanity as we confront the challenges of the 21st century. At a time when humanity is grappling with the impact of illness, such as infectious disease and cancer, scientists are working diligently to develop the methods to contain virus and cure disease. These diseases underscore the critical importance of biopharmaceutical science in our time. The Tang Prize Foundation is honored to have established a prestigious international award in this field to recognize individuals for their outstanding innovations and contributions. Through this initiative, we aim to inspire more people to follow their example and work together for a better future. Today, we are very proud to announce the recipients of the Tang Prize in Biopharmaceutical Science. The Foundation would also like to thank the Selection Committee and its chair, Dr. Zhang Wenchang, for their tireless efforts. Congratulations to all the soon-to-be announced laureates, and I wish you all the best. Thank you.
Thank you, Dr. Chen, for the opening remarks. And now, ladies and gentlemen, I'd like to invite Chair of the Tang Prize Selection Committee in Biopharmaceutical Science, Dr. Zhang Wenchang, to announce the 2024 Tang Prize Laureate in Biopharmaceutical Science. Dr. Zhang. Dr. Zhang, would you please use the microphone? Thank you very much.
Distinguished guests, members of the media, I am now honored to announce that the recipients of the 2024 Tang Prize in Biopharmaceutical Science are Joe Harbner, Svetlana Mosov, Jens Juhl Host, they are jointly awarded for the discovery of GLP-1 7 to 37 as an insulinotropic factor and the development of the GLP-1 7 to 37 based anti-diabetic and anti-obesity drugs.
Thank you, Dr. Zhang, for unveiling this year's Tang Prize laureates in biopharmaceutical science. Now, please join me.
Okay, and here I'd like to briefly introduce these three laureates to you. The first laureate is Dr. Joe Haberner. Dr. Haberner graduated from Santa Ana College and the University of Redlands. Majoring in chemistry, he received his MD degree from University of California School of Medicine. He retired from Harvard Medical School last year. Before retirement, he was a professor of medicine at Harvard Medical School and also was chief of the Laboratory of Molecular Endocrinology and associate physician at Massachusetts General Hospital. The second laureate is Dr. Svetlana Mosov. Dr. Mosov graduated from Belgrade University, Yugoslavia, and received her PhD from the Rockefeller University, majoring in biochemistry. Right now, she is a research associate professor at the Rockefeller University. She was an assistant professor at the Rockefeller University and was an assistant in biochemistry and a member of the endocrine unit at Massachusetts General Hospital, and an instructor at Harvard Medical School. She also was an associate at Harvard Hughes Medical Institute and director of HHMI, Massachusetts General Hospital Core Facility. The third laureate is Dr. Jens Zu Host. Dr. Host received his master and PhD degree from the University of Copenhagen, majoring in medical science. Right now, he is a fellow of the International Union of Physiological Sciences Academy. He is also a senior group leader at Novo Nordisk Foundation Center for Basic Metabolic Research and a full professor of the medical physiology at the University of Copenhagen. Congratulations to these three Tang Prize laureates. Thank you for your attention.
Thank you, Dr. Chen, for unveiling this year's Tang Prize laureates in biopharmaceutical science. And now, please join me to welcome academician Gong Xingjian of Academia Sinica to give us a detailed introduction of the Tang Prize laureates in biopharmaceutical science.
So I'd like to share with you a discovery of an amazing drug that can treat both diabetes and obesity. The number of diabetic patients worldwide exceeds 500 million, and more than 1 billion people in the world are now living with obesity. It is predicted that over 50% of the world will be overweight or obese by 2035. These are huge health threats. Let me remind you about the diabetes and the blood sugar regulation. When we have a hearty meal, we are happy, we are satisfied. But our glucose rises up in the blood, and that needs to be controlled, usually by a hormone called insulin, released from pancreas. What the insulin does is to really to process glucose into the cells so that the cell can generate energy for us, or in liver as glycogen, as storage of energy. In such a way, we can maintain our normal blood sugar level. In diabetic patients, the insulin release usually is insufficient. And sometimes insulin cannot process the sugar into the tissues. As a result, we have high blood sugar with the bad consequences of kidney, cardiovascular diseases, retinopathy, and so on and so forth. So type 2 diabetes is caused by faulty regulation of insulin production and functions. So how is insulin regulated? How does pancreas know when to release insulin? When we have a hearty meal. In as early as 1930s, scientists already proposed there must be some gut hormone, they call it incretin, that is released from intestine and reach the pancreas to induce insulin release. 50 years later, thanks to our laureates this year, we have GLP-1, glucagon-like peptide, identified as one of the incretins. GLP-1 has receptor called the GLP-1R, or GLP-1 receptor, on the surface of the pancreatic beta cells. When GLP-1 engages the receptor, it transmits a signal to release insulin and produce insulin. So GLP-1 is a major mediator of insulin production. What is GLP-1? GLP-1 is a 37 small peptide, amino acid small peptide. It is generated from a larger protein, pre-proglucagon, through enzyme cleavage. The cleaved product, GLP-1 37, however, is not very active. It needs to be further processed into GLP-1 7 to 37, 31 amino acid long, which is now bioactive. The discovery that the GLP-1 7 to 37 is bioactive is very important because now there's a potential to use GLP-1 7 to 37 as antibiotic therapy. In the ensuing 20 years, many scientists begin to work hard to develop GLP-1 7 to 37 as drugs. But that turned out to be not as straightforward because GLP-1 7 to 37 has a very short half-life, 2 minutes in the blood. That cannot be a drug. And this is due to there's an enzyme called the DPP4 that can degrade GLP-1 7 to 37. There are two ways to overcome this difficulty. One is, of course, to make GLP-1 7 to 37 more stable by chemical modification or genetically engineered. These peptides are similar to GLP-1 737, but it's refractory to digestion by DPP4. Therefore, they are more stable. But they still engage the receptor on the beta cell, so they can still transmit signal. So this group of drug is called the GLP-1 receptor agonist, or GLP-1RA. The other way of doing that, of course, is to inhibit DPP4, hence DPP4 inhibitor. Both types of GLP-1 based drugs now are in clinical application. So in 2005, GLP-1 RNA was first approved by US FDA to treat diabetes. This is the first successful application of this discovery. What happened next is totally remarkable. Initially unexpected, but this unexpected surprise is a pleasant surprise. What happened with the patients who received GLP-1 RA not only have their diabetic syndrome reduced, they also lost weight. And in such a way that now we know that the GLP-1RA can suppress appetite, resulting in weight loss. We do not know the exact mechanism yet, but there's some suggestion our brain cell contain GLP-1 receptor, therefore can sense the presence of GLP-1RA and signal to us to reduce our craving for food. Recently, GLP-1RA has also found other health benefits, such as liver disease, kidney disease, and heart disease. They can treat some of these diseases. So in 2014, GLP-RA was first approved by US FDA to treat obesity. So now this drug has two indications, can treat both diabetes and obesity, and has become blockbuster drugs, already benefit hundreds of millions of people. We owe the success to three brilliant scientists, Dr. Hebner, Dr. Mossoff, and Dr. Host, for their discovery of GLP-1 737 as an insulinotropic factor, and the development of GLP-1 7-37-based anti-diabetic and anti-obesity drugs. Dr. Joe Hebner, at the time, at Mass General Hospital, Boston, and Harvard Medical School, he was the first to clone the anglerfish pre-pro-glucagon gene and discover that this precursor protein contains glucagon and another glucagon-like peptide called the GRP. Subsequent cloning of the rat pre-pro-glucagon gene showed that contained glucagon and two additional peptides designated as GLP-1 and GLP-2. And the anglerfish GRP is a GLP-1. In collaboration with Dr. Mossoff, his lab showed that GLP-1 7-37 can induce insulin release from the pancreas rather than the entire GLP-1, and demonstrated GLP-1 7-37 is active in humans, paving the way for clinical application. Dr. Svetlana Mossoff was the first to synthesize GLP-1. She, at that time, was also the director of the Howard Hughes Medical Institute core facility. She was the first to synthesize GLP-1 7-37 and develop several experimental approaches to detect the GLPs, identifying the active form of intestinal GLP-1 to be GLP-1 7-37. In collaboration with Dr. Hebner, she showed that GLP-1 7-37 can induce insulin release from the pancreas rather than the entire GLP-1, and demonstrate that GLP-1 7-37 is active in humans, paving the way for clinical application. At University of Copenhagen, Denmark, Dr. Jens Yu Host independently isolated and identified GLP-1 1-37 and subsequently GLP-1 7-36 amide as an active incretin, characterizing the biology and the physiology of GLP-1 7-37 and demonstrate its therapeutic potential leading to successful drug development. His lab also reported that GLP-1 7-37 inhibits gastric acid release and slows down gastric emptying. Perhaps that has something to do with the anti-obesity potential. In summary, the findings of the three scientists together ushered in an era of GLP-1-based drugs for treating diabetes and obesity. Their extraordinary discoveries laid the foundation for further development by scientists in academia and the industry, bringing in blockbuster drugs with great future prospects. This is an exemplary story of translating basic research into pharmaceutical success with major impacts on human health, which reflects the spirit of the Tang Prize in Biopharmaceutical Science. Thank you for listening.
Thank you. Thank you very much, Dr. Gong, for the insightful introduction. And now, ladies and gentlemen, let us watch a video of the 3 Tang Prize laureates in biopharmaceutical science.
I'm deeply honored by being selected as one of the prestigious 2024 Tang Prize laureates in biopharmaceutical science. I thank the Tang Prize Committee for selecting me for this award. It is very gratifying indeed to receive this peer recognition for my contributions to the discovery of GLP-1 and to see that these contributions are helping to alleviate illnesses in people throughout the world.
I was really surprised. I did not expect to receive the Tang Prize. And I didn't even follow when the announcement for the Tang Prize is going to happen. So when the email I read first thing in the morning, and I actually had to read it twice in order to understand really what was the meaning. But my next reaction was that of really immense gratitude to the committee which awards the Tang Prize for recognizing my contribution to the discovery of GLP-1. I was really, I'm really humbled that I'm put in the company of so many important scientists whose work actually impacted the life of so many people.
So I was very surprised. I hadn't expected this at all. And I don't know how the prize committee actually found me, but I'm of course very, very grateful for this. And I'm really looking forward to coming to Taiwan and receiving the prize. I've been to Taiwan before, and I liked it very much when I was there last. It's beautiful. And I'm really looking forward to coming back.
Congratulations once again to the 3 laureates. Ladies and gentlemen, thank you for joining us at the 2024 Tang Prize Laureate announcement in biopharmaceutical science. And tomorrow we will be making the laureate announcement in Sinology. The Tang Prize award ceremony will be held on September 27th this year. So we look forward to seeing you then. Thank you very much.